We were talking about the common dream of a new generation of natural resources for humankind, so there will be enough for everybody. For us to get there, we need to get around the bottlenecks that we, the SynBio industry, have been caught up in.
Hey, Karl, how's it going?
Oh, man, I am just incredibly tired. I took the red eye in last night after last week's SynBioBeta. I slept about an hour and a half, maybe a little bit more. So my biology is lagging. And those of you who listen to the Grow Everything podcast know that this doesn't happen very often, but Karl needs some caffeine, creatine.
Yeah. I hear you. I'm also still a bit tired from all of the excitement and socializing and learning what we did at SynBioBeta. It was incredible. My brain's still buzzing. I'm in full processing mode. So we'll have to do a whole recap in the next episode. We'll do a deep dive into what we've learned. There were such great themes, everything from hyperscale bio to engineering the immune system. It was incredible. So anything you want to tease before we move on?
The last episode we played was our recap of the 2024 SynBioBeta. And so the next episode after this one will be our recap of 2025. And all I'll say is, even my voice is recovering because we did so much talking. You ran a great panel. It was super well attended. People want to hear about how startups can interact with large consumer product companies. Mm-hmm. And the entire thing is a whirlwind. So stay tuned, listeners. We are going to do a deep dive on SynBioBeta. Erum teased a couple of the things that we saw. What she didn't mention was space biotech, which is also a big part of it.
Oh, don't worry. I have plenty of notes, so we'll dig. But it was really cool to see all of our friends. We made new friends. We saw our old friends. I reunited with Lessa of eXoZymes 3 weeks after I met him in Copenhagen. So it was good to see him because we are speaking with his business partner, CEO, Michael Heltzen of eXoZymes. And I say we are speaking, I meant I am speaking.
Yeah, so I mean, we have done a number of episodes where only one of us interviews. I was not able to be on this episode with Michael, though I did run into Lassa and several members of the eXoZymes team at SynBioBeta. And just as a reminder, eXoZymes is a company that does cell-free biomanufacturing. These are not just alternatives to using cells as factories, but they really represent a fundamental shift in how we unlock biology's true engineering potential. So I'm really excited about this interview.
We did miss you, Karl, and you'll hear it in our voice. I'm like, oh no, where's Karl? But we channeled our inner Karl and we made sure that your spirit was represented in this interview. And it's exciting because eXoZymes has some great announcements. You'll hear Michael go into what's going on, how eXoZymes is developing. It's good to hear unique business models and business strategies. So if you're an entrepreneur in this space, this is a good exercise to build your playbook out. So that's all I have to say. Let's let myself and Michael take it away.
Take it away. Let's go.
Michael Heltzen, welcome to the Grow Everything podcast.
Thank you so much. I'm excited.
Yeah, I'm so happy that you're here. Unfortunately, Karl is not here. He had to travel. So it's just going to be mano y mano. So both of us are just going to talk. I'm excited because I've learned a lot. I hung out with your boy Lassa in Copenhagen a few weeks ago.
Yeah.
And you and I met for the first time at your bell ringing party before you guys IPO'd on the NASDAQ stock exchange, which was super cool.
Yeah, that was pretty exciting.
Yeah. We were at this penthouse, a beautiful skyline of New York, top of the world. So that was really cool experience. I want to talk about that. But before we go into that, let's first set the stage, okay? We talked a little bit about eXoZymes in the intro, but we want to know a little bit about what you do. And for those that are unfamiliar, there's this concept of cell-free manufacturing. And we want to know, why is it a significant development in biotechnology, and what does eXoZymes do?
Yeah, thank you for the question. And I'm excited about the topic here. So we are, like everybody else, super, super excited about the vision and the future of being able to biomanufacture resources. We humankind need a new way of getting resources without depleting nature and without building everything on petrochemicals.
Yes.
So the shared vision of growing everything, really our heartbeat as well. As a company, when we got started as a project at UCLA and the co-founders laboratory there, it was really a SynBio dream of a publication and wow, this is really fantastic potential, but also a realization of people looking at each other. And this is to the soul of our company. It's like, guys, this won't work at scale. This won't scale up. That means it's not going to be real. It means it's not going to have the impact. And if it doesn't have the impact, why are we doing it in the first place?
Right.
And we have some videos online that lets our co-founders explain that aha moment. Halfway in frustration, halfway joking, halfway serious, saying like, okay, guys, let's build the enzymatic pathway that runs inside of the cell, outside of the cell, because fundamentally cells, they don't want to produce anything they don't need. And we also have to be honest with ourselves that a lot of the things we are asking ourselves to biomanufacture actually makes them sick or even die. So of course they're fighting. Of course they're pushing back. Of course they do what we genetically dictate they have to do. And also just to finish that line of thought, often we then end up with a cell solution that maybe produces the compound we want it to produce, but the isolation cost, taking the chemical out of this cell slurry that we have created, is literally more expensive than the value of the compound itself. And from a business perspective, that's just dead in the water right there. We as an industry have been challenged by that, and the guys took a step back and said, what if you could just run the thing we really need, that is the enzymatic pathway, the enzymes taking some starting point, a sugar maybe, breaking it down to very specific molecules and putting those back together insanely precisely, as only enzymes can do. And if we were in control of that process and it could run outside of the cell, it would be more like chemistry than it would be biology. And with chemistry, we mean being an engineering level control of every single thing that happens. And if something breaks, we can go in and measure on it and go like, oh, the molecule pathway stopped right here. So it's this enzyme that isn't doing his job. And therefore, that enzyme needs to be faster or better or survive the conditions. And can we genetically engineer the enzyme with evolutionary force that we calculate via AI to not be that bottleneck, to survive that condition? Can we basically recreate the enzymes, but externally, exo, outside of the cell? So the eXoZymes are AI-optimized enzymes that can survive on their own as a chemical reaction accelerator. And that's how we get the best of both worlds. So it's like chemistry, but without the petro and all the pollution. And it's the power of biology, of evolution, but sped up and controlled. So that's the core of the company and the core of the whole technology platform, and why we are often described as the next generation of biomanufacturing.
Wow. Yeah, that's incredible. Thank you for that explanation. You did a great job of painting the picture and understand the struggle that happens when you're just using cells. So this term eXoZymes is very intriguing. So why introduce this new terminology, and how does it help in communicating your technology's value to partners outside the synthetic biology sphere?
It was really a realization where we ran around in Monrovia, a little bit outside of Pasadena here in LA, where we were excited about the terms we were using, cell-free biomanufacturing, and it was a very, very nice fit for what we were doing until we left the building, a typical startup story where you leave the building and then when you start speaking like that, everybody else was like, yeah, I know what you're talking about, but it doesn't fit with what I know about cell-free. And we were like, oh, cell-free protein synthesis. Mm-hmm. Is what people mean when they say cell-free. And in other circles, I come from the genomics background. When I say cell-free, we mean cell-free DNA that is basically cancer biomarkers. Right. So we were using words where people were getting confused about what we actually meant. So we took a step back and said, okay, despite that we would have loved nobody else used the word cell-free, that's just a little bit too good to be true. We need to take responsibility for the language we use, and we need to get to the core of what it is we do differently. So despite we as a company also use cell-free protein synthesis, and therefore it can very easily be confusing. We took a step back. We had conversation with a lot of our collaborators and people in the industry and said, guys, we need to clean up the language. People are not getting this. And we sat down and based on that, came up with the eXoZymes. So instead of enzymes, eXoZymes, that are the external accelerators of the chemical reactions that enzymes can do so precisely.
Okay, that makes a ton of sense. Thanks for sharing that. And just this whole process, being more efficient, taking this evolutionary developed process and taking it out of the cell that used to do something else, but now we can make all sorts of things. And you recently made a big announcement at SynBioBeta about a new spin out called NCTX that's producing NCTs. Can you tell us a little bit more about what NCTs are, why it's such an important breakthrough, and what makes your platform uniquely positioned to produce it?
Yes, absolutely. It's very exciting for us as a young company coming out with the first use case of our core technology powered by eXoZymes, as we say. NCTX is a company we have spun out around the business opportunity around NCT, a small molecule found different places in nature, but for example in hemp seeds, but at very, very, very tiny amounts, literally trace amounts. So if you want to have enough for a dose of anything, then you're ending up taking down the rainforest scenario, back to the depletion of the natural resources. That doesn't fly. It doesn't fly commercially, and it's almost like a tease from nature. We have this compound that has incredible capabilities and potential from a pharmaceutical perspective, and yet we can't reach it. So of course it becomes, is this something we can build with SynBio? We already know the pathway by sequencing the natural way of producing this compound, and we can lift that genetic information out and say, now we know the code that codes for the enzymes. Let's lift that out. Let's make those enzymes happen. Let's sit down, reprogram those enzymes to remove the bottlenecks, make sure they can survive outside of the cell, all of these fancy things we do with our AI design platform and our basically bioengineering testing that we have up and running. And to cut to the chase, NCT have in preclinical work that have been done by other great people, not us, basically been proven to have a effect of non-alcoholic fatty liver disease. That is a disease that barely have any treatment options today, and is a disease that is more than 30% of all people that has. It leads to the next stage of that disease that is the much worse version of the disease where the liver literally stops working and breaking down. It is one of these diseases we as a generation have gotten ourselves due to basically how we live and the obesity challenge. So we get a lot of fat on the liver, and the liver doesn't know how to shed fat. So despite you lose fat, then you're still challenged. And in all cases, fat on the liver is really a challenge because the liver is the organ in our body that takes everything and sorts it and filters out all the things that shouldn't be in our body. Slowing down that recycling center is really bad for our system. So having a unhealthy liver is a very easy thing to understand why that's bad. And the ability to go in and have a small molecule that helps the liver shed that fat is just a enormous potential. So it has potential from a nutraceutical point of view and from a pharmaceutical point of view. And the difference between those two things is, of course, both a regulatory thing, but also where nutraceuticals will have to be like grass, generally recognized as safe, and be in the more natural product version, where over on the pharmaceutical side, we can make much, much higher purities. And specifically with our platform, we can make analogs. So we can take the natural compound, and then we, with the enzymes, can start building them slightly different so that they have other pharmaceutical features. And this is basically what makes our platform unique, is that we allow for a new generation of small molecules where we design and engineer them instead of before a lot of small molecules was frankly just randomly made, put into a screening library, thrown against a target and hope that it stuck. And then that one molecule that stuck on that day under those circumstances was then further developed on, where this is much more of a design, test, build cycle. We can go over many, many times and understand exactly what do we need the molecule to look like to have the properties, not just binding to the target, but to do the different features molecule.
That's incredible. I mean, I'm really just curious. I think we need to have you back on, just talk about the spin out. I mean, I want to get back to eXoZymes because it's an entirely amazing platform, but just curious how you arrived at NCT, because again, there's so much you can do with this platform. We'd love to hear a story of how you arrived at NCTX.
Yes. First of all, it's one out of many things we're going to do ourselves and together with partners. So it's just the first, but obviously it's not a random first. Because it lends itself very well to this fact that nature have allowed us humankind to see the molecule already. So there's a lot of people that are able to toy around with it a little bit, but then come to a point of, but how do we actually get hold of it, physically get enough of it that it can turn into a product, that it can turn into something that makes impact. We looked at it and we were like, this is going to be a good technology fit with where the technology is today and over to this, and especially this gearing of the natural product version. the way it comes out in nature, that version we can make in very high purity. That in itself is a value if that is the compound someone want to take to clinic. But we can also sit down and make these analogs and say, should we change some of the features of the molecule so it actually targets the specific indication disease? It's a molecule that have shown, we mentioned non-alcoholic fatty liver disease because it's the huge unmet need, but there's actually a number of other things that the molecule have shown beneficial for gut health and other things. At its core, we chose it because there's a lot of people that have shown an enormous interest in it but couldn't get to it. And that's what we see as our mission of unlocking these enzymatic pathways that have gotten stuck inside of cells. People have been trying to do this via cell lines. They've been trying to do this chemically via petrochemicals, and they're trying to isolate it in nature. We see our mission as coming in with a new way of orchestrating biomanufacturing and SynBio, and therefore liberating these enzymes from the cell, building them as eXoZymes, and allowing people to have access to the potential.
Yeah, that's incredible. I definitely want to talk about this orchestration of different partners working together, whether it's NCT or different compounds. But let's talk about the big elephant in the room. I know this has been talked about at SynBioBeta. I moderated a panel about this topic, but it's about scaling in synthetic biology because that has been a common hurdle. And probably because many people might be using just the cells. This could be for other companies that are using cell-free, but how does your eXoZymes platform address this challenge? Do you foresee a future with distributed deployment of your technology?
The last question first. Okay. Yes, we definitely want to empower the world with this core technology. So we want to build biosolutions for people together with people. So when we say powered by eXoZymes, it's a reference to the original Intel Inside phrase was actually powered by Intel. And it's literally that reference to come get a core solution and build your reality around it so it becomes the perfect fit for your market, your problem. So it becomes your competitive advantage. Obviously, like everybody else introducing something new, we want to show how it works and not just show up with a piece of technology and waving that around, but showing how the technology can be translated into a competitive advantage, a biosolution for specific fields. We have a number of things coming up. It's so relieving for us to now be able to talk about at least the first ones, but we have more coming up. Back to the dream we were talking about, the common dream of a new generation of natural resources for humankind so there will be enough for everybody. For us to get there, we need to get around the bottlenecks that we, the SynBio industry, have been caught up in. And you pointed it out already, scaling have been the bottleneck. And you can be really crude and say like, oh guys, if it only works in prototyping and academic paper levels, but it doesn't work at scale, is it then even real? Mm-hmm. Is it like a dream that doesn't have a chance to become reality all of a sudden becomes something negative? So getting really deep into why is it? We can get the cell to take in the DNA, we insert into it, it expresses the enzymes, the enzyme pathway starts working inside of the cell, but there's so much in the cell over the whole history of evolution, at least 3 billion years, that have been about be as efficient as possible. So it has all of these regulatory systems that tries to downregulate or turn off anything it doesn't need. And we just have to accept the core thesis that despite the cell is a fantastic development environment where we get a lot of support for all the systems that are up and running, that is not what those systems are there for. They're not there to service us the way we tried cell-based, and then having to accept that, take a step back and say like, okay, we actually need to take care of everything ourselves if we want to be in this engineering level control where if something changes, it's us doing it and it's not a living cell, because a living cell will always be fighting for survival, will always be fighting for generating the next generation of itself, and that doesn't include what we're making it do. And as we talked about, you then start ending up in all these problems of sick cells and cells doing things. Literally, you're fighting your own platform at this point. Wow. You're right. It's been a conversation that has been uncomfortable to some degree for us as an industry, but now at least we have a solution because our technical co-founders dove deeply into that. What does it actually take to run enzymes outside of the cell? And I'll be frank with you, the first time I heard about it, I was like, you can't do that. Yeah. That's BS. There's no way. That literally goes against everything I know. So I had a pretty strong reaction. But I've taken that back because they were thorough enough to sit down and say like, okay, what are the support systems, the energy, the cofactor, all the kind of support system that needs to be in place? Let's rebuild them so we are in engineering level control over every single thing that happens in our reaction. So there's no living matter in our reactions. There's therefore nothing randomly happening. If something breaks down, it's either us that got the recipe wrong, or it's a bug from the perspective not of a living bug, but in the code of us coding the enzymes. It also lends itself very well to my personal journey career-wise, where I started in bioinformatics and basically have seen the generations of better and better engineering level control we have got now. So I would say this is the newest generation of AI and design and us using knowledge to basically simulate evolution. Okay, now we know what it's going to take genetically to get to this point where the enzyme can do what it couldn't do outside of the cell because it will fall apart, or it couldn't take the heat, or it couldn't work without a cofactor available. And we're just like, okay, let's be in control of the cofactor recycling. So we recycle. We have enzymes taking care of the enzymes. We have enzymes regenerating the cofactors. It's complex technology. Again, it's just the natural version, but with engineering level control.
Yeah. And I can imagine like traditional fermentation or even precision fermentation is often produced in batches. Does your cell-free manufacturing have a continuous manufacturing capability?
It depends on the compound we're making, but yes, it's much easier to make continuous. But we frankly often get so high titers that we don't really need to go in that direction. Continuous manufacturing is typically if you go in the direction of trying to make it cheaper. When you think about it, precision manufacturing, how precise can it be when it's literally alive and it's literally fighting? I really respect all the people working really, really hard out there. But when people get to the point where they start realizing they can liberate the enzymatic pathway from all of that by taking it outside of the cell and reconstituting that as something where they just take decisions, and then it doesn't change again because there's nothing alive there. It's just chemical reactions with the power of nature.
I love that you're paying attention to the words, and you said you had the responsibility of the language and precision. Is it really precision when you have other technologies that could be even more precise? I appreciate that you're being very thoughtful, especially for us at Messaging Lab. You're all about words and messages. So I really appreciate that.
Just to double click on that, the whole eXoZymes, sure, it's the name we put on the company, but that's actually more recognizing that's the kind of biosolutions we build. The word eXoZymes, we have not trademarked. It is for everybody to use. We have released a peer-reviewed open publication on it together with all our partners. People are free to say eXoZymes as a way of saying cell-free biomanufacturing and confused with cell-free protein synthesis. That is also a fantastic technology that is really good at prototyping, but again, at scale really taps out. So eXoZymes is for the scaling part.
Right. And can you just paint a little pictures, like how big is your manufacturing capability? Because we talk a lot about distributed manufacturing here on the Grow Everything podcast. And if Karl was here, I'm tapping into my inner Karl, and he would talk about modularization of factories and putting them next to maybe more downstream processing if required.
I can almost hear his voice as well.
Yeah,
So yes, well, I do listen to your podcast. So let's address that question from Karl that we are proxying here. Let's start with being a bit humble here. We address the smaller in volume business opportunities first, because despite what I've just said about scale, this is not easy. This is not straightforward. If you start talking about some of the biggest use cases that I hope and foresee out in the future. And I'll exemplify this. So what we're working on first here is the nutraceuticals that also have pharmaceutical potential. Some of the partnerships we are negotiating and co-developing on, there will be a world market that doesn't need more than maybe some kilograms of API, the active component of the pharmaceutical ingredient. And that's easy. We can do that in our current lab. We run up to 100 liters. And with the titers, we get a kilo of active pharmaceutical ingredient out of that.
That's good.
It is, but it's a drop in a bucket if we start talking about sustainable aviation fuel.
Oh, yeah.
So let's take that example, because we happen to have a project where the DOE came in and said, you have such a different technology platform that where the cells doesn't get sick. where you're not really battling the whole sickness and isolation challenge a cell-based platform has. And they were basically, we want to make isobutanol, that is the precursor for sustainable aviation fuel.
Hi, a quick shout out to our sponsor, Messaging Lab, the force multiplier for biotech. Your biotech company is making the world a better place. You know that, we know that. But does the world? There's a big reason why some biotech companies attract investors, sign up customers, and get attention. That reason is strategic communications. At Messaging Lab, we translate complex science and economics into compelling narratives. And we have done that for the most successful biotech companies across healthcare, agriculture, personal care and beauty, materials, and the list goes on. We're here to make sure your ideas not only get heard, but resonate with your audience. So if it's time to amplify your company's voice, visit messaginglab.com to explore how we can elevate your story and grow your business.
But the problem is that because that molecule is so high energy density, it turns toxic for the cells. So we're killing our own platform. We can't isolate it without it costs more. Like the whole thing we've talked about a couple of times now. And they basically came and said, like, would you mind trying to boot up your system doing that? We have some significant grants on that from the DOE and the DOD now, pushing that forward. But the reality is that for that to make a big impact on that market, just aviation fuel, we're talking about supertanker after supertanker after supertanker that needs to be sent off worth of product. And that's unfortunately not next week, because that is something that has to be built up in many, many levels. And just the amount of steel in the ground and capital bound to that capex investment and the gearing and all of that, and getting the price point down so that someday it will be the same price or cheaper than petrochemical. Mm-hmm. That's just a long journey. So we do start with scaling with the low-hanging fruits, and that's why it's nutraceuticals with pharmaceutical potential. That is our focus first. But as a platform, I don't think there's a lot of things we couldn't envision ourselves building biosolutions for if there's the right partner and it's the right time and the maturity of the technology have come to the states where we can make it cheap, fast, good enough.
Right, yes. And we talked a bit about manufacturing, steel in the ground, a lot of hardware. Let's talk a little bit more about the software. Your platform leverages AI-engineered enzymes operating outside living cells. So can you delve into how your AI approach generates data and trains machine learning algorithms and accelerates development?
Yes, I know it sounds so complex, and that's because it is. I'll therefore describe it in a more layman accessible way.
Thank you.
There will be a couple of my scientists at home that will be ready to have a strong conversation with me. It's like, that's not exactly.
Right.
But forgive me, if you want to dive into the details, I can geek out on this. I am so passionate about it, but I also know when I start doing that, where people hang up and basically there'll be another podcast I'll rather listen to. So let's dive in.
Yes.
What is amazing is that we have had this development of algorithms since I started professionally. But the first startup we had, there was a bioinformatics startup where we did what we called machine learning back then, but it's the same thing, just more mature algorithms now. It's basically pattern recognition. So can we study the data, see a pattern, lift that pattern out, make it an algorithm, and then rerun that and make a better prediction? And especially if we have a circular system that we call reinforcement training, where when we change something, we predict it should be better. Then if it's better, then we have learned something. But also if it's worse, then we have learned something, then steer in the other direction. This is how your Tesla or other EV with self-driving is driving itself. It's by every single input, it tests the output. Do I get rewarded? Did I do well or did I do poorly? That's how you train algorithms. So how do you connect that to anything like enzymes? Typically, and I'm one of the guys that try to apply these things to genomics and other things. It becomes really, really difficult because that learning cycle is so long and there's literally so much noise in the data collection back to the steering mechanism that it falls apart. What's beautiful about genetic engineering, changing one nucleotide at a time with enzymes, is that one change leads to one feature change. So we can relatively fast make that circle. With that circle in place, we can then start training the system. So we can basically, one change, what did we learn from that? To make that circle fast, people have been doing this for a number of years. They were doing it with CRISPR. They were doing it with big sequencing instruments. We have just a much more nimble system. To take a step back, let me just see if I can make it even more precise here.
Yeah.
So the way we engineer enzymes with AI is basically by taking advantage of that when we change one nucleotide, one base pair, then we can really quickly see, did that improve or make worse what the enzyme is supposed to be doing? And we just do that circle really quick. Actually start by simulating it because there's enough data we have been able to train the algorithms to have a sense of what will be the right one. So we start with something called a zero shot, where we just take like the 100,000 best simulations of what it should look like. Then we press that in the lab. We test those versions of the enzyme up against what it's supposed to do under the condition it's supposed to do it. And then we just feed the algorithm back and learn from that. So that's how we are able to do, Poplar said, like 1 million years of evolution in a day.
Yeah.
And I'm pointing out that it's a day and not a month and not a quarter, because we have systems that are just much, much faster than what anyone else have had access to ever. That's where it's both the power of the AI engines we are firing up and using, but it's also that we have very fast and cheap data generation that is much more signal than noise than anyone else. That's why we're able to push evolution much further than anyone else.
Yeah. So data generation a part of a high throughput wet lab experiments?
Yes.
Of course.
It's taking advantage of that we don't need to express the enzymes inside of a cell.
Yeah.
So cell-free protein synthesis style insights to how we can do these things. It's literally fantastic. From a technology nerd perspective, that I live in this biology is technology, technology is biology world. This is the purest form of that. We're literally coding it on the computer, ordering it with nucleotides being synthesized really quick, really cheap compared to what we had access to 5 and 10 years ago. We insert it in systems that are not alive, and we see the enzymes. We get to sit there and optimize. And then when we have figured out evolutionary style, but simulation style, then we lift that knowledge out and we go out and we put it up in the big system, and it becomes a big chemical reaction.
Yeah, absolutely. I mean, we all live in a simulation, so totally understand that.
You seem pretty real to me. I saw you in New York, and I'm pretty sure it's you. But I know what.
Yes, I definitely am a bot right now. I'm just kidding. No. So, yes, we know speed is a game changer. Your AI-powered development process reduces production timelines, right? So how does this acceleration impact the types of partnerships you can form?
Well, the NCT project is a really good example of that. From the idea and the first people showing interest in it to that we were holding it in our hand in the lab because we'd made it. Well, it was counted in weeks, right? 1/10 of what a normal cell-based system would have costed, if not less than that. And in weeks, not months, not years. It just speeds up everything a lot. Then when you especially feed that into that it's so much faster in the booting up, but it also goes into a scalability system afterwards, then just makes everything much more impactful.
Yeah. So we talked about speed, we talked about 1/10 of the cost, which is checking off all the boxes when it comes to business.
1/10 of the cost in the R&D perspective.
Yes.
Yeah.
Yep. And so you have this commercialization strategy, includes spin out, joint ventures, possibly licensing deals. So how does this approach facilitate multiple market entries simultaneously? And what types of partners are you prioritizing?
So obviously, like any other technology, we have the current technology. We're already working on the next generation. It's actually the different tools that makes up the technology stack where we're pushing each of the tools forward. We will talk about BioClick a little bit later, for example. And then how does that all fit with people's here and now needs? Especially because there's a lot of people that have burnt their fingers on just give me $10 million and I'll back to you in 3 to 5 years with something that works. And then when the day came, there was nothing to show for it. And that's been disappointing for people, very, very understandably. So having things where it's tangible much faster is what we're looking for. And that's why it's the nutraceuticals with pharmaceutical potentials that we're honing in on first, because there's so many things that natural products have already been kind of tried out, maybe even all the way through parts of the clinic, but then gotten stuck in, sure, we could make a little bit of it. We could maybe even test it on patients. It has a fantastic potential. We can't make it in real scale. Gotten stuck there. Those are the projects that deserve to be released from their bottlenecks first. Those are the ones we are prioritizing in partnerships and in self-development.
Right. So you're looking for companies that are making nutraceuticals and perhaps would use NCT to formulate it within one of their products?
So that's a segment, definitely. Yeah. And those are people we're talking to, but some of them find interesting is that they may have, if it's a true nutraceutical that's like, this is the compound found in nature, we want the exact same compound. Then it's a conversation about how much, how fast, and at what price point can it be made, and does it live up to their expectations? And frankly, because we're still a small company, we can't just work together with everybody. Does it live up to our very high bar of being one of these extraordinary going first business opportunities? Mm-hmm. Does it have to wait a little bit until we get our capacities booted up? So that's the one perspective. But the pharmaceutical potential is really intriguing because there's a lot of things nature have already solved, from giving us access to compounds, but they're maybe not potent enough, or they're maybe not pure enough, or they're maybe not pick your side effect, pick your problem, pick your bottleneck. And we can literally sit down and say, like, okay, for example, with BioClick, that's a tool where we can sit down and say, for each of the enzymes that breaks the starting point down and builds it up, those are not random. We can be specific in how things are put together. That's a level of how we build the small molecules, a level of control that humankind have not had before. The power of enzymes, but in human design control. And that means we can make these analogs of the compounds. So that's very much where our conversations are as well, because it just has an amazing opportunity for biotech and pharma companies to sit down and say, we have a small molecule that is good enough, but there's a hair in the soup of some kind. And we can basically let's make that soup, but without the problem, without the hair. That is everything from making the compound more potent, making either metabolize faster or metabolize slower, or all the different things you can do when you small molecule engineer. That is something petrochemical medicinal chemists have been doing for a long, long time, but they have never had access to the kind of precision we offer. That's really where we are starting. That's where you will see the most impact fastest.
Wow. Yeah. So we double clicked on BioClick, but BioClick is available for subscribers to use, or are you using it for your clients? How can people interface with it?
To start with, BioClick is a tool we developed for ourselves. We have even just gotten a grant to take it to the next generation of that tool. For the fellow nerds out there that would like, we put out a fair bit of information on it. So go to our webpage, you can read about it. I won't take it up here, but it's an amazing example of the kind of precision that is now available to humankind when it comes to engineering enzymes, and engineering specifically using the enzymes to engineer the small molecules.
Yeah, that's really cool. Yeah.
It is mind-blowing. We are offering that in the partnerships where we team up with a partner. It's something that can and most likely will be opened up for later on down the road. Right now, it is still complex technology, and we're wrapping our fat fingers around those small steering mechanisms still. It will get so much better just within the next couple of years. And that's probably where it's more relevant to start talking about releasing it.
Okay, yeah. Well, we'll put a link in the show notes about it, so people can double click on BioClick. I feel like I'm so clever saying that, but it's not that cool.
Wordsmithing
is
your
Yes, that's right. Yeah, I'm smithing some words. So let's talk a little bit about life after the IPO. So how has access to public markets influenced your ability to orchestrate partnerships with larger companies compared to when you were previously funded?
What fundamentally have changed is we now, by being listed on NASDAQ, have access to kinds of capital we didn't have access to before. And that means we're in much more serious conversations about being able to gear things up. So when we sit in front of a partner that is maybe literally 1 million times bigger than we are, that's maybe not the exact number always, but they're much bigger organizations. We can still be taken serious because we can go out and not just build technology the world have not seen before with all the opportunities and competitive advantages that can bring, but we can also come with the capital ourselves. So the negotiation power has changed, but also not from a we win over others. It's more like they can actually happen at all, that we can stand behind our own technology and bring it forward. And to echo NCT again, that's a good example. The ability to be taken serious when we go out and spin out a whole other company is because we are a public company, we can be taken seriously differently.
Yes, that's true. You guys are in the big leagues. There's so many exciting things that are happening today. But looking ahead, what are your priorities for the next year? Are there specific commercial partnership announcements that are on the way? Any joint ventures that we should anticipate? Anything that we should keep our ears for later this year?
You should definitely keep your ears out for us. But when that's said, as you just pointed out, we're now a public company. So I need to make sure everybody gets exactly the same information at the same time. So I can't talk too much about the future and what we're going to do. When that's said, we've already promised that this year would be the year where we started showing some of the applications of the platform. And as you know, NCT is the first step of that. There will be more this year. It depends on the negotiation with our partners and some other things that basically is, again, throughput scaling. How many people can we work with at the same time? Does our partners even want to be public about that they're working on it? Maybe they would like to surprise their competitors in their specific markets in regards to what they're working on. And that limits how early we can talk about things.
Yeah, that's cool, because that was a test question. So you succeeded. I was just testing you to see if you'd answer it.
Yeah.
And so everyone, he passed the test. He stayed true to the public markets and his shareholders. Congratulations.
Yeah, I do my best. The SEC is keeping a good eye on guys like myself. And I also think it's important that we as an industry now realize that we're in this next stage of, we can't just be toying around with prototypes and versions and being early stage. It's like, this is real business now.
Yeah.
It's therefore serious. And as a public market CEO, I take it very serious that I know there's a lot of people out there that is investing us and supporting us that way. And from that perspective, we want to live up to not just fair playing rules, but also delivering on what we say. I will be less over-promising. It will be all about promise we want to deliver on.
Yes. Yeah, that's great. We'll see that hockey stick shaping up for everyone. I'm very excited for you guys. This NCT sounds amazing. Can't wait to see that get into the market, get into the hands of the people. And I'm excited for the future of the pharmaceutical world, especially because there's so many medicinal properties in so many plants that are out there that are just living in a plant. And there's all this research. We could just get our hands on it, and then we can produce it using eXoZymes. It's going to be a great future for us. So I want to take this time to thank you, Michael, for coming on the Grow Everything podcast. This has been very illuminating. I'm very excited for you, Lasse, and the team, everyone there. Alejandro, special shout out to you, son of Karl. But thank you so much.
Yes, thank you so much.
Erum, I know you missed me so much during that interview, but tell me, what did you think? I have a lot of thoughts on that interview, but what are your thoughts on your interview with Michael of eXoZymes?
It was a great interview. I'll have to say, I enjoyed talking to Michael. He is a great communicator when it comes to talking about the business of the technology of biotech. What does that mean? How do you think about it? I really like the way he communicated the technical problems. So he said the cells struggle during fermentation. They can either get sick, meaning contaminated, or there's challenges to isolate cells or molecules, which is interesting when it comes to downstream processing. So the way that he framed some of the concepts when it comes to using biotechnology or leveraging enzymes, it was great to listen to. Because one of the things he also said was we have to be responsible for the language when it comes to talking about biology and biotech and business. As we get more people involved in this industry, we need to be responsible for how we communicate, which for you and I, that is a great message at Messaging Lab.
Yeah, that's our bread and butter. Yeah. And listening to the interview, I really enjoyed hearing him also frame the story. One of the things he said was, we make so much using petroleum products, and it is one of the themes that we talk about here at Grow Everything. And there's not enough people out there helping the world understand that there are other ways of building. Biology is nanotech that works. We can control it. It is everywhere on this planet. We're on a carbon-based planet. We can use that carbon to create new things. And the technical challenges of that, Michael did a really good explanation of how that works. So I really appreciated that.
Yeah. And then the big thing is this announcement about NCTX. We teased it before we recorded that episode. We learned about it during SynBioBeta. That's a great model. It's the spin out to prove to the industry you can make it at scale model. Try to make it a nice snappy term for that. But it's cool. I just think it's great that they have the resources in order to show that, hey, look, there's this very unique molecule. We know that it's found in trace amounts in this plant. Let's amplify it using biomanufacturing. And in this case, it's cell-free. There's different flavors of cell-free manufacturing. We can talk about that another time, but it was cool to hear how Michael approached it, taking all the bits that work in a cell, take it out of the cell and make it into this like a biochemical synthesis pathway, which is cool to think about from an engineering standpoint for engineers out there that are not trained in biology. It's good to know that you have these gears and pulleys to make the thing. So I like that. I'm excited to see how the nutraceutical industry will receive NCTX. I hope they receive them with open arms. And I'll just say that one thing was that NCT could be an Ozempic supplement, which sounds very lucrative. Just in terms of maybe it should be GLP-1 supplement because then you're not isolating Lilly here. But it would be cool to see if that catches fire in terms of being an actual supplement that people do use at scale, at the scale of the GLP-1s. But what did you think about this announcement?
Yeah, this idea of having supplements that you take when you're on a certain medication makes a lot of sense. And I think I had one conversation with someone who was taking a GLP-1 that mentioned that there are sections in Whole Foods or health food stores that really put all the nutrients that you're supposed to take together on the shelf so that people who might be on these medications are supplementing and getting the right kinds of nutrition that you're supposed to get when you take these. One of the things that I heard about GLP-1, I just want to mention this, is that you can get sarcopenia, which is muscle loss when you're on a GLP-1. I want to explore that a little bit because I think it's really interesting, the side effects. But it does seem like the health effects are much greater than the side effects. So it's something that we should dig into. But I just want to make another comment about what Michael said in addition to the NCTX information. Their platform, the eXoZymes platform, again, demonstrates why biomanufacturing isn't necessarily going to be bigger tanks. So those bigger tanks are important to drive down the costs. But really, it's like these smaller, smarter, modular, distributed manufacturing systems that can be local and they can start small and scale as needed. In cases like with the NCTX, you don't necessarily need to scale a lot. I mean, one of the things that Michael made clear, and something that we talk about a lot on the podcast, and that is that there's not enough room for us to grow all the plants that we need. And we've had a number of people come on and talk about using biomanufacturing to get rare actives out of plants and grow them at scale. So I just wanted to comment that I appreciate that. And this NCT news, I think, is tremendous.
Yeah. Well, you did hear that we channeled our inner Karl for that specific question. I'm like, we cannot end this interview without talking about distributed manufacturing. This has to happen. This is Karl's big North Star for this industry. I know you have a variety of North Stars as general direction, but that's a big one. But I'm glad you mentioned that because it's a good way for, I think, also the CEOs and the leaders of these companies to continuously think about, especially when it comes to how much money they're going to raise and what kind of partners they have and how do they partner with companies that have biomanufacturing capability or cell-free enzyme manufacturing capability and are able to take some of their downtime and use it to make batches of product that have a certain value. And I recommend all of our listeners to listen to our interview with Ilan Sobel of BioHarvest, because this does dovetail into that conversation about biomanufacturing and how to create ingredients. And the difference between BioHarvest is that they have a direct-to-consumer product. So again, there are different business models for biomanufacturing when it comes to nutraceuticals to pay attention to. It'll be interesting to see how eXoZymes and these other companies segue from nutraceutical industry to the pharmaceutical industry, which is way more lucrative for businesses to partner with and could be more beneficial to pharmaceuticals. I know we'll be paying attention to it. If you know of any companies that are doing a good job of going back and forth between the two industries, first company that always comes to mind is J&J. Janssen as underneath J&J, and now they have they rebranded their consumer division to Kenvue, and they all work together on the higher level. I know I worked at Janssen. I did not meet the Kenvue people, but anyway, I'm digressing. It's just interesting to see how one company can serve multiple markets and how two markets are very closely related. One's more highly regulated than the other, but it's cool to see the dynamic between the two.
Yeah. Well, I think that was a great interview. Good job, Erum.
Thank you.
And thank you, Michael Heltzen, for appearing on the Grow Everything podcast. Do we want to tease out anything else for next week's episode?
Yeah. As we said at the top of this episode, we are going to do a recap of SynBioBeta 2025 next week. We're going to get into it. I took so many notes. Karl and I kept meeting. We're like, we got to talk about this in the podcast. Everything from space biotech to reprogramming cells, writing biology, writing genomes. It is mind-boggling, but we're going to bring it to you in a nice, comprehensive, snackable way. Listen, stay tuned. Thank you for listening to the Grow Everything podcast.
That's the pod.